material for today. So what we're talking about is pregnancy and lactation. Oh, there was an interesting, so you may have noticed that there's been an interesting question of female reproductive endocrinology in the news lately with the athlete Caster Semenya. Does this ring a bell at all? Oh, there's a woman race sprinter, I think, maybe 400-meter runner, who she's been winning, races since 2009. And what's in the news is that there's been questions about her womanness. She has high levels of testosterone, and she doesn't look like the Western patriarchal ideal of a woman. So there have been some issues going on here, right? And so they are now calling her intersex because of her high testosterone level and her appearance. And this is horrible, right? So because her appearance does not look like what People magazine says a woman should look like, and because her testosterone levels are higher than typical, they're now forcing her to take testosterone lowering drugs in order to compete as a woman.
What's her name?
Caster Semenya.
Is that she lost her case?
Yeah, that's right. She was appealing the ruling. She lost her ruling. She lost the appeal, and so she's being forced to do things that other athletes are not being forced to do. It's outrageous. And so someone could say, but she has levels of testosterone that aren't in the range of a typical female. Well, you could say the same thing about Michael Phelps. His hands are outrageously big. His feet look like paddles. You could say he doesn't have a body of a typical male, and they didn't strip him of his awards, right? I mean, yeah, he looks like a man. He has the People magazine, archetype of a man, and yet he has this phenotype of a body that makes him look like a seal in some ways. When you think about the size of his feet and the size of his hands, the length of his arms, and nobody wanted to take away his malehood or his humanhood because he's got this sort of strange human body. So it's pretty outrageous. And but now you know Semenya's high levels of androgens, where they come from, cell type, what cell type might not be converting them into estrogens. I'm just pointing out that something that might allow you to be thinking deeper about a news story than your peers out there. Okay, that was completely aside. Let's talk about pregnancy and lactation. So what we're going to do is we're going to start here at ovulation, where there's the LH and FSH surge that leads to ovulation of the oocyte with the corona radiata, and then we'll talk about, so this little thing here is supposed to be a sperm fertilizing a cell, fertilizing an egg, and then cell division, implantation. I'm going to go into this in detail, or a little bit more detail, implantation, but the key thing is the release of chorionic gonadotropin from the implanted uterus, in the fully developed uterine lining. Once it detects implantation of the embryo, releases chorionic gonadotropin, which sustains the corpus luteum, which allows the corpus luteum to continue to develop throughout pregnancy, maintaining high levels of estrogen and extremely high levels of progesterone. Remember, this is 1,000 times higher than this. So that's what we're talking about here, a ovulation event that results in a pregnancy. And then we'll talk about the endocrinology of lactation. So ovulation is the sort of expulsion of the oocyte and the corona radiata from the ovary. And it is, I think I showed you this slide before, it is picked up by the fimbria of the fallopian tube, and then it sort of sticks, attaches, and then cilia within the fallopian tube propel the oocyte, the egg, into the fallopian tube to the uterus. The egg is viable for only a day after ovulation. Sperm can survive in the female reproductive tract for a couple of days. So there's a narrow window for fertilization. Fertilization usually happens in the fallopian tube. If you may have heard of a thing called ectopic pregnancy, where a fertilized egg implants in the fallopian tube, not in the uterus, because of a block of the fallopian tube that prevents the egg from moving, but doesn't prevent the sperm from getting there, and doesn't prevent fertilization and development. And so an ectopic pregnancy can be dangerous, right? Because now we have a developing embryo implanting in the, or trying to implant in the fallopian tube where it doesn't belong. So let's switch gears just a little bit. So sperm, there are obstacles to fertilization placed in the female reproductive tract to sperm and fertilization. The vagina has an acidic environment, which is not, healthy for sperm, so that kills some of them. There is a mucus block of the cervix until the sort of perfect timing of fertility. So sperm that gets in the vagina might not get into the uterus because of the mucus plug at the cervix. And then there are white blood cells within the uterus that recognize the sperm as invaders and try to kill them off. But nevertheless, 300 million sperm are deposited. Half of them that get into the uterus. So some of them die because of the acidic environment in the vagina, or don't get into the uterus because of the block at the cervix. Half of the ones that get into the uterus and survive the leukocytes that are there go up the wrong way. They're not very smart. They don't have detectors to say which way is the egg. They just go, and so half of them go the wrong way. So only about 2,000 out of the 300 million actually reach the egg. And you may have heard that one sperm fertilizes the egg. Well, that's true, but if only one sperm cell reached the egg, you would not get fertilization. You need the 2,000 or so to get fertilization, and then one of them typically gets to contribute its haploid genome to the egg. So it only takes a few minutes for the ejaculated sperm that survive and reach the egg to get there. It only takes a few minutes, but it takes up to 10 hours for the egg to actually get fertilized once those 2,000 sperm reach there. So that takes some time. And sperm the egg is viable for about 1 day, and the sperm is viable for three or so days. There is a process called capacitation. So the sperm, so here is a cartoon from your textbook, I think, of the sperm with the head, which is really just a repository for the haploid genome, and some mitochondria that fuel the ATP necessary to spin the flagellum so that it can swim. And the head of the sperm cell, so here are so the mitochondria are actually at the junction between the flagellum and the head. This is what's making the ATP that spins a motor that drives the flagellum. the head is coated with cholesterol, which is a protective layer, and then the acidic conditions in the vagina, which are killing some of the sperm, are actually capacitating. So it's both a plus and a minus. The acidic environment removes the cholesterol, capacitating, that is, giving the sperm the ability to fertilize, but also killing some of them. So it's a trade-off. And then when the acidic environment removes the cholesterol layer, we now have what's called an acrosomal membrane at the acrosome at the very outside of the head of the sperm. And the acrosome, we'll see in a minute that the acrosome is important for the binding of the sperm with the egg membrane, which is the first step for penetration and fertilization. So there is a process called capacitation, and it includes removal of the cholesterol protective layer of the head of the sperm. And this is happening, obviously, in the vagina and uterus. So fertilization, this is an image of a sperm. You know, this is just giving you a sense of the size of the sperm head compared to the size of the egg membrane. But it's never just one. It's hundreds or thousands working to try to fertilize this egg. So here is a cartoon that describes the acrosomal reaction and fertilization. So here we have the egg. Let's talk about some of. So this is the corona radiata of the egg. Here is the zona pellucida of the egg. Here is the egg itself, and so there's a membrane. The egg has a membrane. It's a cell. The egg cell membrane is here. There's a space between the egg cell membrane and the zona pellucida, a very narrow space, and then outside of it is the corona radiata. These little purple things trying to fly in here are the sperm cells. And I think I don't know if they're to scale, they're close. So here is the head of the sperm. The motor machinery and ATP synthesis machinery here, spinning the flagellum, propelling it. The first thing they reach is the corona radiata. When a capacitated sperm meets the corona radiata, there is a acrosomal reaction, which is basically the release of a package of enzymes from the sperm that is degrading the cells of the corona radiata and trying to penetrate through this layer. So the acrosomal reaction is required for the sperm to sort of get to the egg membrane. So here is a sperm releasing its acrosomal enzymes and, degrading some of the corona radiata cells. Here is a sperm cell releasing its so some other group of sperm sort of work this pathway through, and then this lucky guy gets to release its acrosome onto the zona pellucida. But it fails to reach all the way through. Here is one that the acrosomal reaction actually penetrates the zona pellucida, and then the sperm head fuses with the egg cell membrane. So this is the one that makes it all the way through and fertilizes. Now this story here is the continuation of this story. So now we're moving in space, but what looks like we're moving in space to a different sperm, we're just moving in time. So what happens when a sperm cell fuses its contents with the egg cell membrane, the egg detects that. There is actually a release of calcium. So here's a calcium signal. And the calcium triggers what's called cortical fusion. So there are these vesicles. They're cartooned here in green. They're not green, but they're cartooned in green. Cortical vesicles which contain enzymes. So the sperm triggers they also contain calcium. The sperm triggers the release of a cortical granule, which then triggers the release of cortical granules from neighboring cells, which triggers the release of cortical granules from neighboring cells, neighboring granules, triggers release of cortical contents from neighboring granules. We're inside one cell, not cells. So I want to make that clear. One sperm cell fuses with one egg cell. The cortical granules inside one cell have a wave of exocytosis. It's a calcium dependent. It's a calcium wave and then a wave of exocytosis, which goes around the entire egg cell membrane. So I'm trying to use some language that should remind you about the wave of depolarization that is the action potential. I'm trying to just use some language that could help you on the final exam. Calcium dependent fusion of the cortical granules. So you should be thinking of calcium dependent. I wonder if it's synaptotagmin dependent. I wonder if it's snare dependent. The answer is yeah, it's all of that. So again, trying to use some of the language for the final exam. In any case, this cortical granule secretion wave changes the property of the zona pellucida and the space between the zona pellucida and the membrane. Enzymes do their thing. I don't even know exactly what enzymes are released, but there's this wave of exocytosis, enzyme release, changing the properties of this space and of the zona pellucida, preventing more sperm from getting through. So one sperm and the cortical granules trigger this global change around the entire cell, because it's propagated from this one spot throughout the entire cell, to prevent polyspermy. And I guess this changed space has a name called the fertilization membrane, but I don't know that space, that term. Yeah.
So when one sperm goes through the zona pellucida and that space in between, that reaction that is happening there at the top happens throughout the, around the whole
Right.
cell.
It's a
So
wave
then
of exocytosis.
Okay.
Calcium triggers release of triggers the exocytosis.
So it goes.
Yeah, exactly.
Okay.
And it only takes a few seconds.
Okay.
A few seconds for this wave of cortical exocytosis to occur. Other questions? Yeah.
How does the calcium get into the egg? Is it through where
So
the
the
sperm was?
fusion of, so there are, calcium, how does the calcium get in? It's not a voltage-gated calcium channel, I don't think. It's, the release of the contents of the cortical granules releases calcium. It's sort of like intracellular release from an endoplasmic reticulum. Okay, so now we have, this is from your textbook, the flow chart of fertilization. Many sperm get, 1,000, 2,000 sperm get there, bind to receptors, undergo the acrosomal reaction. So they move through the zona pellucida, but one sperm makes it all the way through, the first sperm makes it all the way through, binds to the plasma membrane. Then things happen. The secretory vesicles, or this is another name for the granules. The egg releases contents, this wave of granular release, preventing, making the zona pellucida block polyspermy. The sperm is drawn into the egg, and that detection of this fusion event, along probably with that wave of cortical reaction, triggers this egg to undergo the second meiotic division. So remember, now we have we now have the haploid cell. It has gone through the second meiotic division. It has generated another polar body, but the point is now we have a fertilized haploid cell. until the nuclei of the sperm and the nuclei of the egg unite, and now we have the diploid cell that will undergo embryogenesis. And then there are enzymes that facilitate embryogenesis. So this is a flow chart that tries to illustrate the same content as this slide here. So once the fertilized, so let's say, yeah, so here's the sperm nucleus, here's the egg nucleus. It has not yet fused to form one nucleus, but this is a fertilized egg. It starts to undergo development. So in the few days, so here 30 hours, it's a day later, it's in the 2-cell stage, then 4, 8. By 3 days, it has left the fallopian tube and is now in the uterus and in plants. This whole thing here, going from a fertilized zygote to a blastomere phase, to a morular phase, to a blastocyst, this is developmental biology. Wait, a whole nother course. So I won't test you on the terminology of developmental biology. But you should know that development happens, right? We go from a single cell. It's an amazing story that is worth a whole course of going from a single cell to this body that we have, or even just the body that an infant has, right? Or even the body that a embryological, we don't even have to be in mammals. We can go into insects or echinoderms, right? The development of all animals from a single cell to their body plan is really a remarkable story. The point for this course is that the blastocyst implants in the uterus. That implantation into so remember what's going on here. This is all happening pre-pregnancy, so it's happening during the luteal phase of the female reproductive cycle. The corpus luteum is releasing estrogen and progesterone, sustaining and maintaining the highly developed uterine endometrium, anticipating this event of implantation. Now we have implantation, release of the chorionic gonadotropin, which continues to sustain the corpus luteum throughout the 9 months of pregnancy, and then with continued release of estrogen and progesterone. So the corpus luteum secretes progesterone and estrogen. Remember, 1,000 times more progesterone than estrogen. Progesterone and estrogen stimulate the cell and growth of the endometrium. That's just what's happening during the luteal phase. And then we have an implantation of a zygote, or of an embryo, of a blastocyst, chorionic gonadotropin, sustaining of the corpus luteum. The embryo develops. The amniotic sac is actually embryological in origin, not maternal. And so it develops to encase the embryo. Again, we're not going to talk about this. This is developmental biology primarily. But just so you know, the embryonic sac contains fluids as well as urine from the fetus as when the kidneys start to develop. And it's a protective layer. Both the fluid and the membrane itself protect this pretty fragile embryo. The development of the amnion, that is to say the amniotic sac and the amniotic fluid, again, it is fetal in origin. And so there is a connection between cells that are fetal in origin. So what is this supposed to illustrate? Here is the myometrium of the uterus on the outside. Here is a cartoon of the endometrium of the uterus. This is maternal cells. And this is supposed to be the layer of the amnion, which is fetal in origin. And so there are these structures called chorionic villi. So the chorion is a structure of the amnion that contains amniotic fetal blood. And the chorionic villi are the respiratory, circulatory structures that allow nutrients from maternal blood to enter fetal circulation and allow metabolites from fetal development to be released into maternal blood for excretion. So the chorionic villi during pregnancy are the structures that allow oxygen to enter fetal blood and carbon dioxide to leave fetal blood. Allow glucose to enter fetal blood and allow carbon dioxide to leave fetal blood. Okay? So there is actually this sort of pools of maternal blood that surround this fetal chorion. And so here is a cartoon from your text that shows levels of hormone in the months after of pregnancy. Here is the spike of human chorionic gonadotropin. So this is the spike that triggers and then this is the level that maintains the corpus luteum. The corpus luteum releases estrogen and progesterone. And once again, there's no scale here, which is disturbing because progesterone is 1,000 times estrogen, and I think this is tiny compared to these. So this is sort of a normalized. This is a graph that is normalized to its own maximum. But the point is, a spike and then a low level of chorionic gonadotropin maintains the corpus luteum, which then continues to secrete estrogen and progesterone. Lots of progesterone, 1,000-fold less, but still lots of estrogen, all the way up to delivery, at which time it plummets. All three of them just drop off. And so you can imagine this change, this dramatic change in endocrine levels after pregnancy can trigger a lot of changes in the mother. I mean, so you may have heard of postpartum depression, and some of that is biological in origin because of the dramatic changes in hormone levels. So during pregnancy, estrogen and progesterone levels are increasing. Throughout pregnancy, so the uterine endometrium is highly developed to maintain the, to allow the chorion from the amnion to continue to get nutrients. And the myometrium develops, this very muscular, smooth muscle of the uterus continues to develop during pregnancy. And throughout pregnancy, well, not throughout, but starting at about 30 weeks, so 3/4 of the way through pregnancy. Pregnancy is about 40 weeks. So, into the third trimester, there are weak contractions of the muscle of the uterus. Okay, so now I'm using a word that should trigger some thinking in your minds. Contraction. How do muscles contract? Right? In this class we have only talked about skeletal muscle, not smooth muscle, but the mechanisms are very much the same. We will talk more about smooth muscle contraction in AMP2 because it's more important in AMP2 than it is in AMP1. But you should be remembering, oh yeah, contraction. I should be thinking about what stimulates contraction, calcium, what the calcium binds to, what that does, what that does, what that does, what that does to cause contraction, right? I don't want to go through it, I mean, I will if you ask me to, I'll try to remember, but the point is you should be learning this stuff for the final.
And that could
Contraction.
be your question on the final.
It could be a question on the final.
As you can
It combines what we're talking about today and stuff that you have learned. Happy to answer questions, but maybe it's better for you to just take this alert and review. So there are some contractions. They're weak. The mother may not even feel them early on. They're so small and weak and infrequent. And in fact, the mother could interpret them as baby kicks instead of contractions of the uterus. The smooth muscle cells of the uterine myometrium are connected to each other through gap junction ion channels. So connexin, so this is AMP2, all the smooth muscle cells in the uterus are connected to each other. And so if one part starts to contract, that contraction sort of spreads to neighboring cells. So the point is, the smooth muscle cells, unlike skeletal muscle cells, are connected to each other. And now we're going to start to see some endocrinology. Oxytocin from the posterior pituitary, which is starting to get stimulated during the changes in pregnancy, and prostaglandins, which I'm not going to test you on, but is another sort of endocrine, paracrine signaling molecule, start to develop. Start to increase in concentration, spikes of both of oxytocin and prostaglandin happening, and these small, weak contractions of the uterus. But eventually the fetus gets large. The large fetus presses against the cervix, and that cervical pressure is detected and triggers there's a positive feedback loop here going on. So let's start here. Let's start here at the bottom. Cervical stretch. Why stretch? A big fetus. 8 pounds, 6 pounds, 5 pounds, whatever. Pounds of human being are pressing against the cervix, the mother's cervix. And that is detected. Okay, let's think. Stretch. What could be activated by stretch? Stretch activated channels, right? So now I've just triggered another set of memories, good or bad, in your brain to go and refresh. Cervical stretch is detected. Nerves, right? The uterus is innervated, and so information from the stretched uterus can be transmitted either by endocrine signaling or by nervous signaling to the brain, which remember is connected to the hypothalamus, which is connected to the posterior pituitary, and we have release of oxytocin from the posterior pituitary into the blood. Oxytocin is a stimulant of uterine contraction. And so now we have contraction against a large fetal head, and that causes more stretch. And so you can see here we've got a positive feedback loop going. As when the stretch gets big enough, we have enough of this positive feedback loop to lead to labor and delivery. In addition, the uterus releases a paracrine endocrine signaling, prostaglandins. Prostaglandins are what are important for this uterine contraction. During the female menstrual cycle, the cramps are a spastic contraction of the uterine myometrium, and that is also triggered by prostaglandins. And so drugs like Motrin or ibuprofen are prostaglandin inhibitors. They are anti-inflammatory, but they're also anti-prostaglandin molecules that lower the level of prostaglandins and are important for, reduction of cramping. Well, obviously now we're not talking about cramping, we're talking about labor and delivery. So the stretch directly positively, I'm not even sure what the mechanism of this is, but I do know that the mechanism of this is endocrine and nervous signaling to the brain to start to release oxytocin. Some of you who have ever worked in a labor and delivery environment, you may have heard of Pitocin, and they put sometimes to induce labor. So if a woman goes to say 42 weeks and is still not going into labor, and they're concerned, you know, that the fetus is getting too big, or you know, they want to initiate labor, they can start an intravenous drip, an intravenous application of Pitocin, and Pitocin is oxytocin. So that's trying to get this positive feedback loop going. Yeah.
What happens to the amniotic fluid when it's like super late, you know, I don't remember yet?
So the amniotic fluid, so the amnion usually breaks before labor and delivery. The water, maternal water flow, and that can also be a stimulant for labor and delivery. So the amniotic fluid is usually released in the process, maybe just at the very beginning of the process of labor and delivery. This is an interesting story for me to tell. My daughter is pregnant and she'll be delivering in July. So I've never taught this material when it was so impactful before. She's at about 30 weeks now, so no, that can't be right. She's just starting her third trimester, so she is feeling kicks, but probably not feeling contractions yet. She sent me a video of her belly sort of pumping. It's pretty interesting. Okay, so now we're going to switch to lactation, which is a response to labor and delivery. Well, actually, it's a response that happens before labor and delivery. So the estrogen that happens monthly during the female reproductive cycle and is sustained during the pregnancy, that's an important hormone. And as far as lactation, it does all kinds of things, changes the brain, changes the body, changes the density of the bones of the hip. It changes lots of things in preparation for pregnancy and labor and delivery. But one of the things it does is develop the mammary glands. So estrogen is an important signaling molecule that leads, remember, estrogen is a steroid. Estrogen binds to intracellular receptors. Estrogen intracellular receptors cause changes in gene transcription. In the mammary gland, it leads to development of the mammary gland because of those changes in gene transcription. Progesterone, another steroid, another set of receptors, which increases during each menstrual cycle but is now sustained during pregnancy, leads to so we have the mammary gland overall, and the progesterone is specifically targets what these are called secretory alveoli. Those of you who have taken AMP2 know that alveoli in the lungs are the tiny little sacs that do gas exchange. Well, this is not gas exchange, but they're alveoli, tiny little sacs, which are going to be responsible for producing milk. So estrogen develops the mammary gland. Progesterone facilitates or triggers the development of these secretory alveoli. And prolactin, finally we're getting to prolactin. Lactotrophs are now finally in the picture. Prolactin stimulates the production of milk by the well-developed secretory alveoli. So now remember, prolactin, lactotrophs, they're under negative control by dopamine. Dopamine from the hypothalamus. Remember, dopamine is both a neurotransmitter and a hormone. In this case, dopamine released into the portal circulation at the median eminence is a negative controller of prolactin secretion by lactotrophs. So throughout a woman's life, until after birth, there is high levels of dopamine keeping the prolactin secretion low. But at birth, and specifically at the beginning of breastfeeding, there is a dramatic inhibition of dopamine release that allows for a dramatic production of, or dramatic release of prolactin from the lactotrophs of the anterior pituitary. And now we have prolactin-stimulated milk production by the progesterone-stimulated secretory alveoli. So let's go through this. Baby suckles on the nipple. Mechanoreceptor stimulation. So what am I triggering here? Mechanoreceptors. You should remember there are those corpuscles, remember those in the skin way back at the beginning of the somatosensory system we talked about. So this is touch pressure stimulation of the nipple. So this is not, I mean, it might be painful, but that's not the stimulation that's going on here. We're talking about touch pressure stretch stimulation of the nipple due to the baby. I never asked you to remember the name of those corpuscles. Remember there was Pacinian and Meissner's and some others. But remember that there are touch receptors in the nipple. Those touch receptors are long sensory cells, so they fire action potentials, and that leads to a neural input to the hypothalamus. The hypothalamus receives signals from the skin of the nipple to say, ooh, I've got some stimulation going on. That stimulation is what triggers the decrease of dopamine, which causes the, so you can just cross out these question mark ones. Decrease in dopamine leads to decreased dopamine in the portal vessels, leads to increase in prolactin secretion by the lactotrophs. You want to know the cell name here. The lactotrophs of the anterior pituitary releases prolactin and stimulates the secretory alveoli of the mammary gland to make milk. I just said this whole story again in a flow chart rather than in a cartoon formation. The neural inputs to the hypothalamus also cause the posterior pituitary to release oxytocin. So oxytocin was a trigger for labor and delivery, but it's also a trigger for ejection of milk. So what is it? Myoepithelial cells. These are not exactly muscle cells, but they are epithelial cells that have some contractile properties. Myoepithelial. Muscle epithelial. They're not muscles, but in any case, there are cells that squeeze the secretory, there are cells, it's probably not cartooned here, there are cells around the secretory alveoli that are now full of milk because they were stimulated by prolactin. There are cells that squeeze and help to eject the milk. So the baby's stimulus of the nipple triggers this response, but the baby is not only, I mean, the baby is sucking, but it's not just like when we suck through a straw, which is the sucking is not the only way milk is getting into the baby's mouth. It's also being ejected by the mammary gland into the baby's mouth. Some women report that when they are near term, so almost mothers or highly pregnant, or even after labor and delivery, just the sight of a baby can cause this response and cause their breasts to eject milk. Doesn't happen with everybody, but it's not an uncommon response. Just the sight of a baby can trigger milk production and secretion.
But nothing is happening mechanically.
Even if nothing is happening mechanically. So let's see. Suckling stimulates oxytocin, got that here. Inhibits dopamine, got that here. Okay, so now, it also inhibits GnRH release. So breastfeeding inhibits GnRH release, which is going to keep LH and FSH levels low, is going to prevent any spiking type of behavior of LH and FSH, and is going to prevent ovulation. This is why breastfeeding is a pretty good form of birth control, right? Because no more ovulation can happen as long as there is a baby stimulating this cycle. And so breastfeeding, what? It's, you know, months to years is the range of how long babies breastfeed. There are some women who breastfeed until the babies are walking, so they're not even babies anymore, they're toddlers. That happens. That's a choice that people make. Many women, tire of it after a few months and they start to try to wean. Right? There's a broad range. Depends on the society and depends on the person. You know what? So I've got a bunch of other slides that I could use, but the physical adjustments during pregnancy, we're going to talk about respiration, we're going to talk about changes in cardiovascular system. All of these changes only work really well if you've had AMP2. So I'm going to stop here. I'm just going to stop. We've got, I'm happy to answer questions, but I'm also happy to just give you an extra half an hour to do all the things that you have to do at the end of classes. So let's just stop. We're not going to take a break. I'm going to answer questions, but if there are no questions, we're done for the day. Okay, remember my notes at the beginning of class about the final exam, about the evaluation, about the opportunity to write an essay for Brain Explorers if you didn't go to Brain Explorers, etc. Okay? And I'll see you either later today or next week. No lab tonight, of course. You all know that there's no Thursday night lab.